Hello, I'm Sarah Pizzuto with Trefoil Therapeutics, where we're focused on developing novel therapies to address ocular diseases for which current treatment options are limited. Today, I'll be presenting on proliferation of human corneal endothelia in organ culture stimulated by engineered FGF1 derivative TTHX1114. Here are my financial disclosures. This research was funded by the National Institute of Health under the NCATS TR&D program as well as the Counteract program. Fuchs' corneal dystrophy results from the progressive loss of corneal endothelial cells, or CECs, at an accelerated rate, preventing the endothelium from pumping fluid out of the cornea, which leads to loss of visual acuity. The current therapeutic approach is transplantation of either the whole cornea or decim its membrane, both of which come with the risk of rejection and lifelong steroid use, as well as a number of additional complications. An alternative pharmacological treatment that would stimulate proliferation of CECs could restore pump function of the endothelium, giving patients their sight back with a far less invasive approach. But can CECs really proliferate? While it's long been thought that CECs do not divide meaningfully in vivo, likely due to arrest in G1, ex vivo studies using donor corneas have demonstrated expression of the proliferation marker Ki67 in corneas stimulated by injury. In addition, CECs derived from both normal and diseased corneas have been shown to divide readily in dissociated cell culture. A limitation to many of these studies is their short-term nature. A common study design is the pulse chase experiment, where cells are briefly exposed to a radioactively labeled DNA analog and then tracked over time to follow the fate of the cells that incorporated the labeled compound. But since proliferation-competent cells may only represent a small subpopulation in vivo, the odds of catching them in division during a short pulse are slim. For this reason, studies with longer exposure times to labeling agents are needed to further characterize the proliferative potential of CECs after the first round of division. You may recognize fibroblast growth factors, or FGFs, as a standard component of cell culture media, but these endogenous proteins are also known to be both protective and potently mitogenic for CECs. FGF1 has specifically been shown to stimulate CEC proliferation and accelerate endothelial recovery. However, the application of FGF1 as a therapeutic is limited by its structural instability, resulting in a short plasma half-life. Amino acid substitutions to the FGF1 molecule can increase thermal stability and reduce susceptibility to proteolysis. These engineered FGFs have extended biological half-lives and are potent accelerators of wound healing in the skin. This study sought to explore CEC proliferation in the human organ culture system using both wounding and pharmacological stimulation by the engineered FGF TTHX1114. In these experiments, normal corneas were obtained from Lyon's eye bank and incubated in growth media containing 8% fetal bovine serum for 24 hours to recover from storage in Optisol. Corneas were then cut into quarters as shown on the left, and each piece was cultured for either 2 or 7 days in low serum media containing 0.4% FBS, and with or without TTHX1114 at a concentration of 100 nanograms per ml. All quarters were incubated in EDU, a thymidine analog that's incorporated into the nuclei of cells that are actively synthesizing new DNA, and can later be labeled using a fluorescent azide and click chemistry. This allows us to observe and quantify the percentage of DNA synthesizing cells and use this as a measure of proliferation. After 2 days in culture, quarters were fixed and stained with Hoechst 33342 to visualize nuclear DNA, which you can see here in blue, Alexa 488 to visualize nuclei that incorporated EDU, shown in green, and an anti-Zo1 antibody to visualize cell borders in red. In the undisturbed midzone, the observation of evenly distributed CECs with Zo1-rich cell junctions and typical endothelial morphology show that structure is retained in both control and treated quarters. At the cut edge, we see slightly less regular cell boundaries with increased cytoplasmic area, but this is to be expected with the disruption of contact inhibition caused by quartering. As you can see in the upper panels, both control and treated quarters exhibit low levels of EDU incorporation in the midzone, whereas at the wound edge we see much higher levels of EDU incorporation. Additionally, edge quarters treated with TTHX1114 demonstrate a larger amount of EDU incorporating cells compared to the control. We quantified this observation in 10 normal corneas by counting the total number of cells and those incorporating EDU in the mid and edge zones separately. In the mid, 7 of 10 corneas showed higher EDU incorporation in treated quarters compared to control, increasing the average from just under 2% to about 4%. However, this observation was not statistically significant. At the edge zone, treatment with TTHX1114 increased EDU incorporation in 9 of 10 corneas, with an average of 18% compared to 10% in the control, and this observation was significant. It's important to note here that EDU incorporation in these corneas is highly variable in both the stimulated and unstimulated conditions. Next, we wanted to explore longer-term proliferative capacity of CECs in organ-cultured corneas. 4 normal corneas were quartered and cultured using the same methods as before, but 2 of the quarters were taken out to 7 days with continuous exposure to EDU, while the other 2 were fixed after the first 48 hours. Of note here are the clusters of EDU incorporating cells in 7-day quarters, as opposed to the pairs of presumably daughter cells that we frequently see in 2-day quarters. The largest distinct cluster size at 7 days was 8 cells, suggesting that CECs undergo a complete cell cycle in about 2 days, which is consistent with the cycle time observed in cell culture, as reported by Nancy Joyce. There were no noticeable differences in CEC morphology between the 2 and 7-day quarters, suggesting that corneas can be maintained in organ culture for at least 7 days under these conditions. Quantification of EDU-positive cells in these corneas showed that in both the mid and edge zones, the number of cells incorporating EDU at 7 days remained small and did not appreciably increase after an additional 5 days in culture. The panel on the upper right shows the mean percent EDU incorporation of the combined mid and edge in control quarters. The average EDU incorporation rate was 6.8% at the 2-day time point versus 7.5% at 7 days, which again was not statistically significant. Looking at the panel directly below, we see that treatment with TTHX-1114 resulted in an increase in EDU-positive cells from 8.6% at 2 days to 14.5% at 7 days. This difference also was not statistically significant, which may be due to the small sample size of only 4 corneas. In addition, it is possible that EDU incorporation could hinder further division, which, if true, may explain why some corneas did not show more labeled cells at 7 days, as seen in the mid and edge bar graphs. Furthermore, the difference in response between the 2 pairs here reflects just how high the variability between donors is. But taken together, these results, along with the observation of dividing cells in contiguous groups, support the possibility of proliferation-competent CECs that, when stimulated, may undergo multiple rounds of division. However, it's clear that more research is needed to confirm these findings. One of the quarters from the previous experiment, which was treated with TTHX-1114 for 7 days, was imaged in series at increasing depth of focus, and the stack of images was compiled into the GIF on the right to show CECs migrating transversely down the cut edge. You can see here that many of these cells have incorporated EDU, further demonstrating the additive yet independent response to wounding and treatment. Studies by Patel and Sinu have suggested release from contact inhibition upon injury as a potential mechanism of CEC proliferation, while stimulation by TTHX-1114 likely operates via FGF receptors on the CECs. By the same mechanisms, epithelial cells from the anterior side of the cornea would be expected to migrate up the cut edge and potentially interfere with our analysis. These cells, identified by their own unique ZO1 pattern, were occasionally observed but were not counted. So to conclude, we demonstrated that corneas can be maintained in organ culture for up to 7 days without obvious visible change in CEC morphology, and treatment with TTHX-1114 had no apparent phenotypic effect. Using this model, we showed that treatment with TTHX-1114 for as short as 2 days stimulates CECs to proliferate at a low level in the undisturbed midzone, as well as at the edge, where drug and injury combined produce a cumulative yet independent effect on CEC proliferation. Corneas kept in culture longer showed higher EDU incorporation, which presents in clusters, suggesting the possibility of a progenitor population, which is also susceptible to stimulation by TTHX-1114. Additional research is required to quantify these clusters and to further characterize proliferative capacity in vivo, but overall our findings indicate that TTHX-1114 may serve as a valuable therapeutic for the regeneration of CECs and thus the restoration of visual acuity in patients with low endothelial cell counts. If you have any questions, please feel free to use the comments feature here or contact me directly via email. To learn more, please watch our other presentation, which can also be found on the ARVO website. We would like to thank the NIH, Trefoil's founders and advisors, our lab staff, collaborators, and investors. And here are my references. Thank you so much for listening.